educational videos on biogas by Hycons.
We are back. So, ninth chapter in this series is about the digestion. What will become the gas ultimately? So, you take any organic matter in the nature, ultimately it has to become gas. Biodegradation is all about gas. It may become alcohol or anything. So, ultimately the end result is going to be gas. So you will come across various vocabulary when you are reading on the internet or any other source of science. Generally, people utilize these terminologies, which is very important to understand. One is the dry matter. What is dry matter? Now, if we take 1 ton of grass, 1 ton of grass would have about 75% water. So 250 kg or maximum, if it is well grown, probably 300 kg will be the dry matter content. So excluding the water, what is left over is dry matter. So how it is done? It is very easy. About 105 degrees in an oven, it is kept. There will be still some moisture left over, but still it is called a dry matter content. So that is taken for the test. Typically, in any scientific experiment, the calculations are given based on the dry matter basis because water as such do not have any role in methane production, but it is required for all other purpose of bacteria movement or mixing and those purposes, but water as such has no direct contribution to methane generation. In this dry matter there is some theoretical amount of water is left over, so that is, once that is deducted, it's called total solids. So it is a pure solid we are talking about. So absolutely no moisture, that is called total solid. Then within this total solid there is a ash content. Ash is the minerals, calcium, magnesium, or whatever the minerals are there, that is ash content in the organic material. Then there is a digestible solid. So if you look in the above picture, the almost everything is organic matter, but is it digestible? No, these bones are more of ash because so much of mineral. The coil is organic material, but it's a lignin, so it is considered as not digestible. So we'll have to arrive at deductible, we have to digestible solid we have to arrive at, and that is by deducting whichever is not digestible: lignin, keratin, bones, all those things. So lignin is actually it is digestible through various means, but in a normal case of anaerobic digestion, it is not possible to digest. That's why we usually deduct it. So whatever is not practical, that has to be deducted. So once that is deducted, we are left with volatile solids. So volatile in the sense this can change its phase. It can go from solids to liquid to gas phases. So it is volatile. So this volatile solid is increasable by the action of enzyme, chemical treatment, ultrasonic, heat, and basically what we are trying to do. This indigestible, let us say cellulose, hemicellulose, those fibrous parts. When Hycons does the enzymatic treatment, it will get converted to volatile solid. So we are increasing the volatile solid. That's why we claim a better yield. So ultimately, for gas production, volatile solid is what matters, because volatile solid is going to get converted to gas. The rest, all the things, ash or digestive, non-digestible solids, all those things will come out as a solid fertilizer. But it has no much value for the CBG. So how this take place? There are various fancy jargons: hydrolysis, acetogenesis, acidogenesis, methanogenesis. Many people think this or promote this as if these are all isolated processes, but it is not like that. All these bacteria are working in tandem. It is not a single bacteria doing something and giving to other people. So it is always all these bacteria are together and they make the entire process happen. So first phase is hydrolysis. What it means is there is a complex polymers which is fiber, fats, oils, all those things we are breaking it down and trying to make it simple sugars. That is monomer and oligomers or the acids. So once this is done, the acidogenesis will happen. So basically, the idea here is these sugars are converted to more volatile organic acids. Then acid will become acetate. So acetogenesis will take place. This is where the vinegar will happen. And while this is happening, anything whenever we break some acid, hydrogen will get produced. So for a brief period of time, there is a hydrogen gets produced in the anaerobic digestion, and people are really struggling hard to stop this process here and capture this hydrogen because it is completely green hydrogen. Then comes the methanogenesis. So once this acetate is created, then what happens? This acetate is taken over or eaten by the methanogenesis bacteria. They will convert the acetic acid into methane and carbon dioxide, and some water will get produced. This is the complete cycle of methanogenesis. If you look at here, so for all the practical purpose, it never matters whether it is a press mud, whether it is a sugar, grass, whether it is paddy, whether it is food waste. There is nothing. There is no bacteria has never recognized those things. What they are all doing is, if it is a fat, they will break it to acid. If it is sugars, they will break it to then this one. If the or if it is in acid form, it is broken into acetic acid. If it is acetic acid, it's made to become a methane. So we can feed at this level, you can feed at this level, and any complexity level. So based on the feed material, the chain will take over, and the bacteria will ultimately create the methane without discriminating what raw material we are putting. I think this is helpful to understand. So we set up commercial plants. You can always contact us, and also you can clarify any deeper doubts if you have. Thank you.